Welcome to everybody. In this lesson I will explain you why fungi are very important, powerful tools in soil bioremediation. Indeed, the environmental contamination is really a serious problem of our society, and if we are talking about soil contamination, we already know that there are many physical and chemical techniques that are available for the reclamation of soil. However, this technique has different drawbacks since they are usually very costly and have also a strong impact at environmental levels. So, bioremediation could represent an alternative to this kind of physical or chemical treatment, and we can define bioremediation as any process that uses organisms, usually microorganisms, hence fungi or bacteria, or their enzyme, to return the environment to its original conditions. To be more precisely, many microorganisms are able to degrade different persistent organopollutants, and hence, but it's really important to understand that we can have a bioremediation by means of different mechanisms. Indeed, sometimes fungi are able to carry out a so-called mineralization, which means that the contaminants are used as a carbon source and are completely metabolized to water and CO2. In other cases, fungi can also perform a so-called co-metabolism, in which the contaminants are not used as the main source of carbon, but they are transformed at different levels. And finally, several times, fungi or bacteria can also impact on the mobility of the absorption of the pollutant. And this is the reason why it's really very important every time to follow also the potential degradation of the contaminants by means of ecotoxicological test. Indeed, as you can see in this figure, the fate of the organic pollutant in the soil can be really very different. We always hope that the microorganisms are able to decrease the toxicity, but we have to be sure of this, since in several times there can also be an increase of the toxicity of the system. So now let's come back to the fungi, to their features. Usually the fungi are characterized by the presence of the hyphae, which is the typical structure of fungi. An hyphae can be defined as a long tube which is essentially able to grow only at the apex, and this tube is surrounded by a cell wall, which is really very important. In the older part of the hyphae, there are starvation compound or compound that can be used for the reserve, but the active part of the hyphae is the tip. One of the features of fungi which make them really very important in soil bioremediation is that hyphae are continuously growing organisms, and these hyphae can only grow at the apex in the so-called zone of extension. To give you an idea of the growth rate of fungi, the fastest fungi grow at about 40 micron per minute. So the hyphae are the vegetative and assimilative structure of fungi, and thank you to the hyphae, fungi can really colonize every part of the soil, and they are also able to penetrate solid substrate, and this is really very important because of their competition with bacteria. So they have access to specific niches which are found in the soil where bacteria can't grow. From a practical point of view, we can divide fungi in two main groups. The more primitive fungi have very simple hyphae, which are called cenocytic hyphae, in which we cannot find regular septation, whereas in the more evolute group of fungi, like Ascomycota or Basidiomycota, we found the so-called septate hyphae, where the hyphae are regular divided by septa in sort of, let me say, cells. These hyphae, as I told you, are really very important for the exploitation of fungi in the soil, for their colonization of soil, making them strongly competitor of bacteria. But what about the nutritional behavior of fungi? From a theoretical point of view, almost all organic material can be used by fungi. For sure, easier is the compound, more easier is the possibility to exploit it, but we have to keep in mind that fungi are the organism which in nature can really degrade the more recalcitrant compound like lignin, keratin and chitin. And and this is really very important because for using the same enzyme, they are also able to degrade xenobiotic, so the main pollutant we have to fight right now. Almost all of you know the concept of rhizosphere, which is the area of the soil which is influenced by the root of the plants. Usually you don't know the term mycorrhizosphere, which is indeed very important because you have to imagine that from every roots, fungi colonize a big part of the soil by means of the hyphae. And from this hyphae, the fungi release enzyme and other nutrients that support the life of our organism, also at a great distance from the roots of the plants. And this is really very important in the so-called mycorrhizosphere. Indeed, the mycosphere concept is really important for the bioremediation, since, as we have already said, fungi account for the up to 75–80% of the soil microbial biomass. In the soil, fungi are able to tolerate extreme environment in terms of pH, temperature and water activity. And on the contrary of bacteria, they do not require water film for their dispersion. And as I try to show you through the concept of mycosphere, the hyphae grow all around the soil, forming a network which is really useful for the transport of nutrient and also for the pollutant. And this is the concept of the fungal pipeline in the soil. But they do not only transport nutrient and pollutant, but they can also transport bacteria all along the soil, and this is the concept of fungal highways. So in this figure you can see the different kind of persistent organopollutant that fungi are able to degrade in soil. Some very important group of pollutant like explosive, dyes, pesticides, polycyclic aromatic compound. These are only a few examples of the molecules that fungi are able to destroy. And as I told you before, the main mechanism involved in the degradation of these contaminants are the same enzyme that fungi usually produce to degrade lignocellulose in nature. In many cases, fungi are really able to mineralize this pollutant, hence transforming this pollutant in water and carbon dioxide. Of course, it's not always so easy to get this result, but now with the new technology, there is a really great result in this field. But why we have to use fungi in bioremediation? First of all, we have to keep in mind that they are ubiquitous organisms that can be found in both aquatic and terrestrial environments, and that they are naturally organized to degrade this kind of contaminants. Through the mycosphere, we have already shown that they have the ability to penetrate the soil, and so they are able to reach the pollutant autonomously. And finally, the enzyme used for the degradation of these contaminants are usually extracellular, and this means that fungi, respect to bacteria, are able to tolerate a higher concentration of contaminants. And moreover, these enzymes are really very powerful for a biochemical point of view in the degradation of these pollutants. Finally, but very important from an economical point of view, we can deliver fungi in soil using very cheap carriers, which are usually lignocellulose material, which improve the capability of fungi to degrade these contaminants. Hence, from a practical point of view, if you want to select some fungi for bioremediation purpose, you have to select strains which are endowed with strong degradative capability towards these contaminants, and this means that they have to produce different kind of oxidative enzyme, like, for example, lactases or peroxidases, but they also have to produce biosurfactant, which are small molecules which facilitate the degradation of the contaminants. You have to keep in mind that you have to select strong organisms able to compete, to fight with the autochthonous microflora present in the soil. And finally, you have to select fungi which can be easily cultivated and that can grow on low-cost carrier. We are not fighting between fungi and bacteria, and so we have also to consider that it's usually much more better to try to find fungi and bacteria which work together, because indeed in natural environment, fungi, bacteria and also roots of the plants usually form consortia, which are physically and metabolically interdependent, and this means that they have already selected organisms which like to grow together and help each other in the degradation of the soil contaminants. So please keep in mind to use fungi and bacterial consortia in your next trial, and thank you very much for your attention.